Metformin's hidden side. The drug your diabetic uncle takes every morning might be making cancer cells kill themselves, or it might be saving normal cells from dying when they shouldn't. Same drug, same dose. Opposite effects. And nobody fully understands why, or how to predict which one is actually happening inside your body right now. So the question becomes, if metformin can push cell death in two completely different directions depending on context, what context actually matters? Here's the thing about medical science. We love a clear answer. A drug that kills cancer? Amazing. A drug that protects healthy tissue? Equally amazing. But a drug that does both, switching between them like a light switch nobody can see. That's the kind of mess that keeps researchers up at night, and that's exactly what metformin appears to be. Metformin is not some exotic pharmaceutical. Over 100 million people take it every year. It's the frontline treatment for type 2 diabetes, dirt cheap, been around since the 1960s. Doctors prescribe it like water. But here's what's been nagging at researchers for a while now. People on metformin don't just get better blood sugar control. They seem to have lower cancer rates. They seem to live longer, oddly long, and the glucose lowering doesn't fully explain it. The question that drove this research was fundamental. Is metformin actually fighting cancer in some hidden way? And if so, can we weaponize that? But more urgently, if metformin can kill cells, is it potentially harming the healthy ones too? Before this study, scientists had pieces of the puzzle scattered everywhere. They knew metformin hit multiple biological pathways. They knew something called ferroptosis, basically iron-triggered cell death, was involved in everything from cancer to heart disease to neurodegeneration. But they didn't know how metformin actually navigated that landscape. The research team set out to solve the riddle of context. The study came from Istanbul. A collaboration between three major institutions: Hamidiye School of Medicine at the University of Health Sciences, the Graduate School at Istanbul Medeniyet University, and Istanbul Atlas University. The lead researchers were Besli, Ercin, Kalkan, Cakmak, and Çelik. It published in July of 2026 in the journal Pharmaceuticals Basel. These weren't theoretical physicists. These were cell biologists who spent years poring over how metformin behaves in different tissue types. And what they were about to find was something that would flip the entire conversation about this drug upside down. So here's what they actually did. Think of it like this. Imagine you have two different neighborhoods. One neighborhood is full of cancer cells. Another neighborhood is full of healthy cells. The researchers essentially walked through both neighborhoods at the molecular level, watching metformin do its work, measuring everything that changed. More specifically, they examined how metformin interacts with a cellular death pathway called ferroptosis in malignant cells versus normal cells. Ferroptosis is an iron-dependent form of cell death. Unlike apoptosis, which is the standard programmed death most cancer drugs trigger. Ferroptosis is like rust happening inside your cells. Iron accumulates. Lipids oxidize. Cells break down. The whole system collapses differently. In cancer cells, they tracked what metformin actually did at the molecular level. In healthy cells, they did the same. They measured protein levels. They looked at gene expression. They watched what happened to iron storage, antioxidant defenses, and mitochondrial function. Some of this work involved direct experimentation on cell cultures. Some involved analyzing existing data about how these pathways connect. The methodology combined biochemical analysis with computational modeling to build a complete picture of context-dependent effects. Here's what they found. In cancer cells, metformin turns into a ferroptosis accelerator. It activates something called AMPK, which is like a cellular energy sensor. It shuts down mTOR signaling, which normally helps cancer cells grow without limits. It reduces something called SLC7A11, which is basically the cell's defense against iron toxicity. It cranks up ferritinophagy, the process where cells eat their own iron storage. It hammers mitochondrial complex 1, causing stress. All of this pushes the cancer cell toward the ferroptotic cliff, and then it falls. But here's the twist. In normal tissue, especially stressed or injured normal tissue, metformin does almost the opposite. It activates something called NRF2, which is essentially the cell's master antioxidant defense system. It protects GPX4, which is a critical enzyme that stops lipid peroxidation. It stabilizes SLC7A11, keeping the cell's iron shield intact. It cleans up damaged mitochondria through better quality control. It basically puts the normal cell in a protective bubble. Same drug. Cancer cell accelerates death. Normal cell activates survival mechanisms. The difference comes down to metabolic context. Cancer cells are metabolically different. They rely on different pathways. They have different vulnerabilities, and metformin exploits those differences brutally. Now, what does this actually mean for you and everyone you know? If you're taking metformin for diabetes, this research is legitimately good news. It suggests the drug isn't just lowering your blood sugar, it's potentially protecting your normal tissues while simultaneously making any lurking cancer cells more vulnerable. That's the dream drug scenario. You get benefits on both sides of the equation. But here's where skepticism kicks in. This is mostly mechanistic research. It's showing how things work on a molecular level. It doesn't mean those mechanisms translate to actual clinical benefit in human bodies. Cell cultures behave differently than whole organs. Whole organs behave differently than whole living human beings. We'd need randomized controlled trials to know if this actually changes survival rates or reduces cancer incidence in real people taking metformin long term. And that kind of evidence takes decades. Also, context is weirdly slippery. The researchers identified what creates different contexts, but they didn't identify all the factors that might flip the switch. What if there are tissue-specific factors they haven't measured? What if age matters? What if other drugs you're taking change the outcome? What if some people's genetics predispose them to one effect over the other? These are open questions, which is exactly what makes this research important and incomplete at the same time. The bigger picture here is that we've been thinking about drugs wrong for a long time. We want every drug to be a hero or a villain. Good for sick people, bad for healthy people. But metformin suggests that binary thinking fails in the real world. The same molecule can be a killer and a protector depending on context. And if that's true for metformin, it's probably true for dozens of other drugs nobody has looked at carefully enough. This research challenges the assumption that universal mechanisms drive drug effects. It raises a genuinely hard question. How many other treatments in medicine are simultaneously helpful and harmful, but we're only seeing the average outcome? How many drugs are we prescribing suboptimally because we don't understand the context-dependent effects well enough to match them to the right patients? This is the kind of finding that doesn't give you an answer. It gives you something better. It gives you a framework for asking better questions. The single most mind-blowing takeaway from all of this is that one of the world's most widely prescribed drugs might be working through a completely different mechanism than we thought, and we might be able to weaponize that difference to actually target cancer while protecting healthy tissue. But the real question is this: if metformin's doing this, what else are we taking that's working in ways we've completely missed? Turns out scientists have been busy, and they're not done yet.